Hello everyone. It's an honor to be here today at BITS 10th Annual Science in Society Conference to present our research titled Study of Short-Chain Fatty Acids Produced by the Gut Microbiota and their correlation between colorectal cancer and the Moroccan population. So, as we all know, colorectal cancer is a growing health concern, and while genetic factors contribute to its development, lifestyle and diet play a crucial one. The gut microbiota, particularly its metabolites like short chain fatty acids, is increasingly recognized as a key player in disease progression. So, now let's go through the outline of our presentation. So to guide our discussion, here is the structure of today's presentation. First, we're gonna start with an introduction, then the background or state of the art. Then we're gonna talk about the key goals of our study or different objectives. So we then gonna go through the methodology overview, expected outcomes, so what we hope to achieve, and finally conclusion and perspective. Let's begin with the background. CRC or colorectal cancer is among the most commonly observed cancer represented by an abnormal growth of cells developing in the tissues of the colon or rectum. According to the World Health Organization, in 2022, it resulted in 935,000 deaths worldwide, making it the second leading cause of cancer-related mortality globally. Additionally, there were over 1.9 million new cases, ranking colorectal cancer as the third most frequent type of cancer in the world. The risk of developing this cancer is related to bad alimentary habits, smoking, intestinal inflammatory disease, polyps, genetic factors, and aging. The incidence of CRC has been constantly increasing in recent years, partly attributable to changes in lifestyle and dietary habits influenced by urbanization and westernization. Although international studies have explored the relationships between these bad habits, the gut, and the development of the pathology, a precise understanding of these links remain incomplete. Recent researches have shown that diets high in red and processed meat, as well as diets low in fiber, can lead to an imbalance in the gut, which promotes the production of carcinogenic metabolites. Conversely, fiber-rich diets, such as those based on vegetables, fruits, and whole grains, are associated with better intestinal health and are reduced the risk of CRC due to the production of short-chain fatty acids, which have anti-inflammatory and anti-tumoral effects. The literature then highlights the key role of environmental and dietary factors in the development of the pathology, particularly through their influence on the gut. So these metabolites in particular are the center of current research due to their anti-tumorigenic properties. However, despite growing documentation at the international level, studies exploring these relationships in specific populations, such as that of Morocco, remain limited. So, what about short chain fatty acids? These metabolites are aliphatic organic compounds composed of a carbon chain containing 1 to 6 carbon atoms. They are generally water soluble and exhibit low volatility. These metabolites, produced through the fermentation of dietary fibers by the gut, have demonstrated significant anti-tumorigenic effects, particularly by inhibiting, for example, the epithelium-mesenchymal transition, which is a key process in the acquisition of invasive properties by tumor cells. Short chain fatty acids, by binding also to G protein coupled receptors, act as signaling molecules to influence various biological processes, including intestinal homeostasis, immune regulation, and tumor suppression. Butyrate, in particular, is known for its ability to induce apoptosis and modulate important molecular pathways such as the SMAD pathway by reducing the expression of proteins like SMAD4, which is a key factor in the initiation and progression of colorectal cancer, thereby limiting tumor progression and metastasis. Furthermore, these metabolites have a beneficial effect on the composition of the gut. They promote the growth of beneficial bacteria such as fecal baculum and bifidobacterium, which have demonstrated several anti-tumoral properties in experimental models. Humanized murine models have also demonstrated that soluble fibers such as pectin extracted from plant cells walls can positively modulate butyrate levels, improving tumor responses, even in cases of resistance to immunotherapy treatments. Research on metabolites primarily focuses on certain populations, while in countries like ours, where the traditional fiber-rich diet is increasingly influenced by westernization, available data remain limited. This transition characterized by an increased consumption of saturated fats, refined sugars, and ultra-processed foods could negatively impact the composition of the gut and the production of short-chain fatty acids, potentially increasing the risk of TRC. And data also from the IRC also show although incidence rates in Morocco are lower than in developed countries, mortality remains relatively high, highlighting the need for targeted research to understand and mitigate these disparities. So, why is it necessary to study these aspects in our population or our Moroccan context? So, for several reasons. First of all, epidemiological specificity or epidemiological data, which are remain limited, dietary and cultural diversity, because they could specifically influence the production of our metabolites and the composition of our gut. And few studies have explored how these dietary patterns affect the development of CRC in Morocco. Regional microbial variability, it is crucial to understand the specific characteristics of our gut and our metabolism. Then studies also here examining short chain fatty acids and their correlation with colorectal cancer are scarce or even non-coexistent. And we're gonna try to fill in the gaps in therapeutic and preventive approaches. Study is part of an effort aimed at better understanding and analyzing the correlation between levels of these metabolites produced by the gut, its composition, and the risk of developing CRC. Also, with several specific objectives like examining medical files, identifying the types and variation of short chain fatty acids and samples from patients with colorectal cancer and healthy individuals, also compare these metabolic levels, microbiota composition, and study their concentration to determine their feasibility as diagnostic tools for early detection and describing the dietary behavior of both groups. Our methodology. Our study will be conducted in several oncology centers or university hospitals. Uh, it will adopt it will adopt or consist of three complementary components to enable a comprehensive analysis. First, we're gonna through descriptive epidemiological study by collecting data from medical records. Um then, to achieve the objectives of this research, a combined approach will be employed incorporating advanced biochemical and molecular techniques, a questionnaire targeting newly diagnosed patients, focusing on fiber intake and food diversity, and as I said, a molecular study incorporating advanced biochemical and molecular techniques. These methods will allow for a comprehensive analysis of these metabolites in the gut composition. Molecular analysis involves metabolic quantification in blood and fecal samples using gas chromatography and QRT-PCR. The study anticipates uncovering uh, evaluating of survival rates and main causes of death in followed patients, quantitative and qualitative data on short-chain fatty acids in colorectal cancer patients, confirm or hypothesize that the production of this metabolite is altered, investigate fluctuation in these metabolites depending on the stage of the disease, identification of dietary profiles associated with increased or reduced risk, and detection, of course, of pathogenic and beneficial bacteria. Conclusion: the study aspires to provide valuable insights into the complex interplay between dietary habits, gut microbiota, and short-chain fatty acids in the context of CRC development. By assessing survival rates and mortality causes, we aim to clarify CRC size in this demographic, analyzing quantitatively and qualitatively the metabolites will advance global research on microbiota metabolites, especially in Morocco, and identifying dietary patterns linked to CRC risk and confirming alterations of these metabolites due to dysbiosis highlights the role of microbial balance in prevention. All these insights could guide targeted prevention and therapies, emphasizing dietary modulation's role in gut health and CRC risk reduction while addressing key research gaps. So, thank you for your attention.